don't get me wrong, I am not against secondary analysis. I am very much in favor of doing secondary analysis. Secondary analysis is extremely important for you to understand the results of the primary analysis. Okay, so it's very important to do them. It's very important to do them correctly and interpret secondary analysis are important, and presenting a p-value is very different from interpreting the result only on p-values. And we'll talk about that in a second. All right, so secondary analysis. How you select the participants, how you actually administer these interventions. You may not administer them very carefully as you should, or you don't follow people well, you don't measure the outcomes well, and in the analysis of the data and in the interpretation of results. As mentioned yesterday, right? The reporting has to be transparent in what you did. And so publication and interpretation of reports is very important. And have loss of participants to follow up. And when they participate in the study, they're volunteering to participate. So they're free at any time to decide not to continue participating. And so if they're lost to follow up and you don't include them in the outcome assessment, those who are lost could be different from those who remained in the trial and can give you bias. So you want to avoid attrition bias by completeness of follow-up. I mentioned the case of the cholestyramine study where they got all 3,810 men. You do intention to treat analysis, but you want to understand it. If you do have it, how do you understand it? So we describe things, we look at sensitivity analysis, and dealing with the missing data. So we sort of mentioned multiple times in multiple ways that you analyze different subgroups. And if you have missing data, then you have to do some sort of creation of the missing information. So you make it up by imputation. Many people do, if you are following people and routinely having them come to clinic and you're measuring your outcome variable, like the visual analog score of pain every once in a while, then they missed the last one. Well, you can take the one previous and carry it forward to this one. That's one way of doing it. But if it's so far back, you don't want to do it too much. We do sensitivity analysis. We've talked about looking at the reason why it's missing. Is it at random? Is it completely at random? Or is it informative? So these are all sensitivity analyses that we tend to do. We compare the per protocol and the ITT for that. But here's the question. What if your primary hypothesis is not significant, but some secondary hypothesis turns out to be? That was a question we had yesterday. Ideally, you should have your primary one significant before you even consider the secondary hypothesis outcomes. But sometimes important and unexpected secondary outcome results may be overlooked if you follow that. So it's always important to look at all of these secondary outcomes. And if there are many, you need to adjust for statistical multiple testing on it. The Bonferroni procedure is then you just divide by the number of tests that you're doing to get the adjust the p-value, or you adjust the alpha level. to do it. It's a very simple one, but outcomes are not often independent. They're all correlated because they're usually ones that are correlated to the disease you're studying. So the Bonferroni adjustment is very conservative. And so it treats all outcomes as equal. Other ways of dealing with multiple outcomes is you create a composite, a single clinically relevant outcome. And we talked about that when we talked about outcomes at the beginning. That's important to everybody. You decide on a single effect measure or decide on a single time point if you have multiple time points, like these people that were doing the acupuncture did not do. And this is what I prefer to do is I looked at the multiple outcomes and I'm now interpreting the confidence intervals and not focusing on the p-values. And so this is my recommendation. But caution, these are essentially the same, right? A 95% confidence interval can be used to do the same inference as the p-value, because if the p-value is less than 0.05, then the confidence interval will not include the null value, and vice versa. If the p-value is greater than 0.05, so not significant, the confidence interval will include the null value. So they're one and the same. But as long as you don't use the word significant, and you're saying the range of possible values of the true difference in this group is such and such, and in this group it's such and such, and for this outcome it's such and such, then you're just exploring, you're discussing, you're interpreting, you're not claiming that you've done a test. Because remember, you have not powered the study to do that. And so if you don't find one, you don't say, oh, there's no effect on this one. That's also the wrong thing to do. Now, those are the secondary outcomes. What about side effects? What do we usually do? Most people just don't really do any formal analysis. You don't see p-values for side effects. If you're going to have p-values for secondary outcomes, you should have p-values for side effects too. You have to be fair to things. But that's not of importance, right? Then secondary outcomes are there for a purpose of consistency, and the side effects are there for the purpose of you interpreting the safety. So what you should do with the confidence intervals, and do not only the side effects at a given time point, cumulative at the end of the study, but you may be seeing that there are a lot of ones and they were occurring earlier in group A than they were occurring in group B. So look at it over time. Do really study intervention. So the effect of the intervention varies by levels of this other variable. So you may be interested in knowing that. But it's a secondary effect. You have not powered the study to have adequate numbers of people that exercise a lot versus people that don't exercise a lot. If that was a real interest, you would have done enough sample size in each of those groups to be able to answer that question. But it also helps, and a lot of clinicians say, I want to do this because it may help me identify subgroups for whom the treatment does work better or does not work at all. Okay, but it's a suggestion for may work better and may not work better. So it's a hypothesis generating one as opposed to a hypothesis confirming look. Okay, so it's always should be viewed as hypothesis generating. And so if you're hypothesis generating, why the heck do you have p-values? Again, we shouldn't have p-values here. What are the possible harms of doing subgroup analysis? That you may incorrectly identify and say, this is a group that it doesn't work, or this is a group where it does work, and you're basing it on observed effects that you did not have the power to have tested. You had not even thought that you had tested it. So here's an example of how this can mislead results. And some of you may already know this. This was the VaxGen HIV vaccine trial that was done in Africa, published in The Lancet in 2003. So this is not a trivial journal, okay? And they had 5,000 volunteers who were given HIV vaccine or placebo, followed for 3 years. Overall, there was no effectiveness. They said there was no difference. They really should have said, we don't have enough evidence to reject the null hypothesis. But they said no difference in efficacy between vaccine and placebo. If you look at, they had 5,000, right? So placebo was given to 1,633, 2-to-1 randomization, because they were able to recruit more by saying 2-to-1, the more likely you'll get the vaccine. And the infection rate here, 5.8. The infection rate here, 5.7. No meaningful difference. And even with that large number, it was not statistically significant because the confidence interval includes 0. Okay. Where did they get that efficacy of 3.8? Oh, I think they're doing this is a percent, not an absolute difference of this. This is why I hate percents also. Okay, everybody loves this. You don't know what is the denominator they're doing. So there's no difference between them. So what do? They torture the data. They look at, if we look at whites and Hispanics, we have 5.4 in placebo and 6% in the vaccine. So the vaccine is worse than placebo in whites and Hispanics, but it's not statistically significant. They had a lot of them. They had 4,500, okay? And despite that, this difference of 6% was not. So the vaccine doesn't work in whites and Hispanics. Then they looked at blacks, Asians, and others. And they had fewer infections in Asians than in blacks, Asians, and others under vaccine than they had in placebo. 67% less. Well, it's the difference is that because now they're doing this relative, right? So it's 67% because 3.7, 9.9 minus 3.7 is 6.2 divided by 9.9 is 66.8. Again, it looks like huge difference. If you do relative things, now it becomes very huge difference. So now they're reporting a huge difference here. They put a star and p-value 0.01 they put. Fantastic results. The vaccine works in blacks and Asians. Then they go further and looked at blacks only, and it reduced it by 78% from 8.1 to 2. Now I have only 314 people that have done it, and I've got this fantastic result, p less than 0.02. Then I looked at others, and so now they had all the different ones, but this is a subset of this is a subset of this is a subset of this, and these two are subsets of that, make up that, okay? And then they did another one, participants who received 3 or more doses of vaccine. Now that is a as treated analysis. Regardless of where they went, if they crossed over this and that, they report that. That's who they included here. So they didn't do an intention to treat in this. And then what did they do? This is the president of the company and the vaccine inventor, all smiles. This is the first time we have numbers to suggest that a vaccine has prevented HIV infection in humans. Okay. We're not sure why certain groups have a better immune response, but these preliminary results indicate that a surface protein vaccine that stimulates neutralizing antibodies correlates with prevention of infection. Stocks of VaxGen go up. They make a lot of money on this. And people started saying, well, wait a second. The overall effect was none. You started torturing the data till it finally said, yes, it works in this group. So what happened? 2 months after they published this, critics bashed HIV vaccine trial analysis. Skeptics questioned the subset analysis, and they had to withdraw the paper from the Lancet. Embarrassment, because people do this. What reason they had? Money. Some people may do this and not get caught, but they had such an egregious one, they got caught. Please don't do this kind of stupid things like these people did. So, when you're going to compare, put p-values in subgroups, if you want to, because people say, yes, I don't want to, I will not use the word significant, but I'm using the p-value as a meaningful statistic to inform that if I had postulated this as a hypothesis, it would have been significant. So I'm just speculating, but I'm providing this as to help the reader understand that the difference would have been, right? But I remember p-values are probabilities. They are not in the past. Probabilities are always in the future. But anyway, let's say you do it. If you do that, don't test each subgroup like they did. Test the blacks, test the this, test the that. You test the interaction of the effect, okay? So if you do these multiple tests, you have increased false positive rate, which is what they got. And so the subgroup should be compared using an interaction test if you're going to put a p-value whatsoever. You probably will find that those interaction tests most of them will not be significant because you don't have adequate power to test interaction because you did not plan the study for an interaction. So again, my recommendation, don't do these, just present the confidence intervals and interpret using words. And those words do not include the word significant. They include anything else. We see a notable difference between this group and that group. We see a substantial difference. We don't see a meaningful difference. Those kinds of words are fantastically ones to use, but don't use the word significance. So misuses of subgroup analysis are many, and this is just a study that looked at misuses of it. And so they looked at a bunch of papers and where subgroup analysis reported, yes, in 35, number of baseline factors included. A lot of them included, at least one, but then, even some included at least 5 of them included more than 7. Number of outcomes for subgroups were a lot. Total number of subgroup analysis of a bunch. Statistical methods for subgroup analysis, bunch of p-values. Interaction test is getting better. That was not bad. This was 2000, so who knows if it's improved. Descriptive only was very little. And this is what it should be, okay? And subgroup differences claimed. This is the difference. This is the problem. People say, oh, I'm just exploring. And then they start claiming things. You should not be claiming things, okay? And then in their abstract, they put these results. Why? Because they may not have had any significant results elsewhere, and they need to have